Hello, students. Today, what we'll be doing is working with what we call fluid thioglycolate medium. This is what we're working with. It is going to denote the oxygen concentration of microbes. Now, some things that you need to understand about this. Do not take it and shake it, turn it sideways, etc. Pick it up nice and neatly out of your tray. So yours, you'll put 5 in one of these test tube racks and have them spread out like I have here. I usually do one test tube rack per table. And then when you're finished, put them all together in one test tube rack as a class, and then put them into the incubator. Now, as I was saying, do not shake, turn, tilt this tube. It is very important because then you're going to change the oxygen concentration of the tube, as well as you'll notice that these should have some pink. It's kind of hard to denote in this video, but it has kind of a pink tone to the very top. That is a dye called resassurin that tells you about the amount of oxygen present in your tube. Now, each tube, you're going to take your Petri dish from last week and you are going to inoculate each tube with the corresponding dish. Let me further explain. For example, there are 5 of these tubes and 5 Petri dishes. So you'll have one that's your control. You're not going to do anything with it. You're not going to transfer. You're just going to keep it sealed. Next is nose. Then you have throat, shoe, and the last one would be skin. Now, we have to label these tubes. So there is tape in your cardboard boxes between you and your lab partner. Again, if you're out of tape on your side, just get some from the other side, the table on the other side. You'll need to cut it in strips that are about as long as 2 joints of your finger. Put your name, initials on it, or something that you can identify these as specifically yours, because remember, we are going to put them all together on one rack. So how are you going to know yours from someone else's? If you notice, this one had tape on it. So we're just going to go wrap it around about midway or so. Do make sure that the tape is not going to go all the way around the FTM tube. So if you do 2 joints of your finger, it's going to be a small portion. It won't go all the way around. Make sure to put your initials and then the location of the location from the Petri dish onto each tube. So for example, I would put my initials for me, MVC, and then I would put, for example, let's say this was the nose. Okay. Now, inoculating these dishes, you will take your inoculating loops. You'll have some up at the front. Get one of those and and you will literally approach the top of the auger. Do not dig down into the auger. You guys did a great job of that last week whenever you did your aseptic transfer. So literally take the very top of the loop and just approach the top of the auger. You will get your bacteria. It may not look like it, but you did. Now, what you do is you open the cap. You notice how I'm doing so. Hold the cap. Take your inoculating loop. Go straight down the center, being careful not to hit the sides. and go all the way without hitting the bottom to the bottom. You don't have to shake it. You don't have to stir it. Just dip it down in there. Dip it out. Okay, just go straight down. Let it sit for a couple seconds. Pull it straight out and throw that away. You'll do that for nose, throat, skin, and shoe. So you'll take the inoculating loop, not the needle. Okay, well, you could use the needle, but anyway, inoculating loop or inoculating needle like you used last week. Take it, go straight down, let it sit for a couple of seconds, and then straight back out and discard. Don't sit them on your table. Don't let them hang out anywhere. Please just immediately discard them in the small trash bins or the large trash bin that you have. When you're done with those, you take them, sit them all together. So now that I've done mine, I would sit them all together. In one test tube rack, and then the class test tube rack will go into the incubator. The size of the class, you're probably going to need 2 test tube racks for the entire class, and those go up in the incubator. We're going to go ahead and keep our Petri dishes from this week, and next week we will do the smears, bacterial smears, unless you're in a different class of mine, and I will talk to you more about that in the announcements as far as when we will do bacterial smears and so forth, but that's the next step that we will be doing as well. Those are your FTM tubes. That's oxygen concentration of microbes. And also check your announcements for more information regarding your top hat and any other things that we have going on in lab.